Hello everyone, and welcome to the first lesson that we're going to be doing online, which is on Mendelian Genetics. And if we're going to be talking about genetics, we have to first of all provide a definition. And genetics is the study of the transmission of traits from one generation to another. This is also what you would call heredity, right? You inherit those traits from your parents, from your family. Okay, so another thing that we also should probably go into is a little bit of vocabulary. Again, as we've done this before, some of it might be you have heard it already, and some of it might be new to you. So let's say that we're going to go with genes. So we know that genes are sort of these unit of instructions. They are these sections of DNA that will code for a particular trait. Okay, so these are sections of DNA that code for proteins. And we saw that the general mantra, or the general idea that in our last unit was that one gene will code for one particular protein. Okay, so genes will code for things like proteins, or depending on what protein they're going to code for, you might have different traits. It might also influence patterns of behavior, for example, whether or not you can camouflage, those kinds of things. Okay, now when it comes to alleles, this is something that we mentioned, but we never wrote down. It's sort of a new terminology. An allele is the alternate forms of a gene. So for example, the gene is hair color. And for this particular gene, I can have the brown allele, let's say, and then we can have the blonde allele. Obviously, we are oversimplifying. There's more than two color of hair, but we're going to say, these are the different forms of a gene. You could have attached earlobes versus the free earlobes, hitchhiker's thumb versus a straight thumb. Those are all different alleles for different genes. And we're going to look at gene locus next. So gene locus, this is the location of a gene on a chromosome. And as we discussed in class, we said that mom and dad will have the same genes in the same location, so the same genes in the same locus, but their alleles could be different. Mom and dad are carrying the same briefcase, but the paperwork inside is different. Now, in order to show you that, what I'd like to do is I'd like to add sort of a little picture on here that kind of gives you the idea of, we have a particular locus for a gene. So this is the allele for short hair, allele for long hair, but the gene is coat length. And so for that particular gene, which is coat length, you can have short or you can have long. Okay, and then here we have another allele. We have a locus for coat color, and so we can have white or we can have black allele. Okay, so hopefully that is clear for when it comes to gene locus. Now, when it comes to genetics, the next thing is we're going to have to see that there are several laws of genetics, of heredity, and those laws are due to one particular person. And so we're going to discuss Gregor Mendel. He's also known as the father of genetics. He was an Austrian monk. And at the time, monks were sort of the only people that had an education. They knew how to read, they knew how to write, they knew how to do math. And also they had to have chores. And so what Gregor Mendel's chore was to tend to the garden. And he started doing experiments with the common garden pea. And he did all of these different hybridization, or all these different crossing studies by pollinating different plants. Okay, so that's what he was interested in. I will show you a picture now of Gregor Mendel. So we'll put that in there for you guys to see what Gregor Mendel looked like. So here he is, our father of genetics. And so what he did is he was interested in breeding experiments with garden peas. And he liked garden peas for a particular reason. We're going to see that garden peas have several different traits, as you can see here. So you can have different color, you can have different flower position, seed color, seed shape, pod color, stem length. So you can see a lot of different characteristics by working with these peas. So they had many different traits. To play with. They also had a short generation time. Which means it doesn't take long for them to grow and for you to see where these flowers are or what the pods look like. But probably the best reason why Mendel liked peas is the fact that peas can be self-pollinated. Whoops. Which means you can take the egg and sperm from the same plant. Or they can be cross-pollinated, where you get the egg and sperm from different plants. By using these pea plants, what he ended up doing, see, he established the basic principles of heredity. Okay. So we're going to see what those basic principles are by looking at Mendel's process. So what Mendel did is first of all, he started by creating a stock of what he called pure breeding plants. Okay. Today we would call those purebreds. Okay, so what it means to be purebred. If I have a purebred Chihuahua and I'm crossing it with another purebred Chihuahua, I should get babies that have characteristics of their parents, no outliers, right? I'm not going to get dachshund legs on a Chihuahua, right? Because that's what happens with purebreds. And so generation after generation after generation, you would expect the same characteristics to show up. If I have a white plant, I should get white flowers. If I have a plant with green seeds, they should have green seeds. Okay? He called these plants the P or parental generation. Okay, so that's what he used in order to do his crosses. Every time he would start off with purebreds, and he would either cross them together, self-pollinate them, or cross-pollinate them and see what he would see. Okay, the offspring, sometimes the word that they will use is the progeny. So you need to know that word as well, progeny, that resulted from crossing two parent plants, was the first filial generation. The first filial generation, are also known as the F1 generation. Fili in Latin means offspring, and so that's where the filial comes from. Okay, so it was the F1. Okay, and then when Mendel cross-pollinated or self-pollinated the F1 plants, he called the offspring, you might have guessed it, the second filial generation, or the F2. Okay. So now that we've seen a little bit of the definitions, some of those things that we need to know as we go into it, let's look at monohybrid crosses. There we go. Okay. So a monohybrid cross, this is a cross that involves only one trait. Okay. For example, stem length, okay, in a plant. Later on, we're going to explore dihybrid crosses. What happens if I get a tall plant with pink flowers and I cross it with a short plant with white flowers? But right now, we're just going to focus on stem length. So just short versus long, okay? And let's have a look at some of the crosses that Mendel performed. So let's say that we were looking at stem length, and we have a long plant or a tall plant being crossed with another long plant. So these ones, we said they're going to be our parent generation. And what he did here is he cross-pollinated, okay, which means he used two different plants. Okay, the egg came from this plant, and the sperm came from that plant. Okay. So when he crossed these two particular plants, what he got is all of their offspring was long. Okay, so all of the F1, the first filial generation, was long. Then he took these F1s and he self-pollinated, which means that the egg and the sperm both came from this particular plant, the same one. And not surprisingly, he got, for the F2, he got long plants. Okay, so this was just sort of one of those control crosses that you would do just to see if what you think is going to happen will happen. But he also sort of, in doing this, figured out that, writing this stuff out, it's a little bit painstaking, and so maybe we should get some abbreviations going. But let's see a different kind of cross. So now we're going to go into a different example. So let's say we're going to call this one example 2. Okay, what if I took a long plant and I crossed it with a short plant? Again, I'm dealing with the P generation. Again, I am cross-pollinating this particular one. And what I'm going to get is I got all long for the F1 generation. And then again, I am self-pollinating this particular generation. And I don't know, he was expecting long, but what he got 75% of the offspring was long, and 25% of the offspring was short. Okay. So Mendel started thinking, okay, I can see that my F2 generation would have some long and some short. But the question that he was asking is, where did the short go here? I see it here, but I don't see any short in the first filial generation. So what happened to that? So he proposed 2 ideas. Okay. Number 1 was the law of unit characteristics or inherited characteristics. Which states that traits are controlled by genes. Now, he didn't use these words back then, but are controlled by genes, which occur in pairs. One from mom and one from dad. Okay. If I were to look at the long and short, I could say that for long, those characteristics, those unit characteristics, if I'm going to have long, I could have long like this. I could have long like this. Or I could have short. Okay, and we'll talk about alleles in just a second. Okay, so for these unit characteristics now, what we know today is that big L, why is it capital L twice? And for that, we have to look at the second law, which is the law of dominance. And the law of dominance, what it states is the fact that when it comes to alleles, they come in a dominant and in a recessive form, okay? And when both a dominant and a recessive allele are inherited, only the dominant will show as a trait. So in this case, long is going to be dominant, and short, whoops, I guess I should write recessive, and that's the short one. So since they are inherited in pairs, what we say is that big L, one from mom, one from dad, this is long, but also big L, again, one from mom, one from dad, this is also going to be long because we have this dominant allele right there. And so the only way that I can have a short plant is if I inherited a little l and a little l from both parents, and that would give the short condition. Okay, let's see how that plays out in that previous cross that we just saw right here. Okay, so let's write that out, but instead of just saying L and S, let's see if we can use our alleles. So I had a long and I had a short, and I got a long as a result. And then when we crossed it, we got 75% long and 25% short. Let's use alleles now. So if I had a long, and remember that these are purebred plants. So purebred plants, I think we could write this one as a purebred. Both of its alleles are the dominant long. And this one, well, the only way we can have short is two little L's. And so then if each one of the parents provides an allele to the offspring, then this parent right here, the only thing it can provide is a big L. And then this parent right here, the only thing it can provide is a little L. Now, I have to see what happens when I take this long parent with this selection of alleles and I cross it with itself, right? Remember that this one was self-pollinated, whereas this one was cross-pollinated, right? So what happens when I self-pollinate it? Okay, let's have a look. Now this L could combine with that L, and so I could get big L, big L. I could have this big L combine with that little L, and so then that would give me big L, little L. I could have this little L combine with that big L, and so again I would get big L, little L. And then finally I could have this little L with this little L, and what I would get is little L, little L. Now we have 3 out of the 4 that are long, 75%, and 1 out of the 4 that is short, 25%. Okay. Now, in the next section, in the next class, I will show you how to set this up using Punnett squares, and we will see a number of different crosses and practice from there. Okay. Talk to you later.